Okay, so again, just knowing basic facts can help you answer questions about the membrane. So the outer portion of these molecules that are interacting with water, they must be polar. We have water in the cytoplasm or inside of the cells. So these areas of the molecules must be polar. And of course, then the only area or the only area in which we're going to find nonpolar molecules is more internally in that membrane. And they're not going to be interacting with water, okay? I purposely picked this diagram to show you guys because I want you to see that they can draw proteins different ways. This is just a protein. It's a protein that's embedded in the membrane. And because this protein has amino acids with hydrophobic side chains in this area of the protein, that's why the protein orientates itself where it does. It's just because of its polar and nonpolar properties in different areas of that protein. And we've learned about side chains on amino acids can have polar and nonpolar properties, okay? All right, I'm gonna move on down here to some new information, okay? Now, I'm gonna add another type of protein in the membrane. You guys probably in past classes learned about receptors, transports, enzymes. Let's talk about proteins like these that actually attach one cell to the other. Your cells aren't just flying apart. They're not just coming off of you. The reason why is because they're anchored to other cells. And there's different types of proteins that anchor cells to cells. Okay, so in this area, these proteins form what's called tight junctions. That's a good name for them because we need to keep it tight sometimes between cells so that, for example, like we're taking a look at the stomach and you have acid and you have enzymes in there. You don't want those out of that area of your body. So that's what tight junctions do, is they prevent molecules from leaking out of someplace in your body, leaking out through the gaps between cells. We don't want that to happen in some areas. We also need cells to be attached so they don't fly apart, but we also need them to be able to do this. We need them to be able to stretch but not come apart. So that's what these B junctions are. So these are called those anchoring junctions. They anchor a cell to a cell, but they allow for stretching of the cell part that they're lining. So like your bladder needs to stretch, your stomach needs to be able to stretch, your lungs need to be able to stretch, okay? So the cells that make up those areas of the body, they're gonna have a different type of junction there that allows them to stretch, okay? And then finally, the gap junctions. Well named, if you ask me. When you think of a gap, there's a hole, right? So gap junctions allow molecules to send molecules to nearby cells through those gap junctions. So if you have a gap junction between your cells, then cytoplasm is able to flow back and forth between those cells. So they can actually exchange molecules that way through gap junctions. So it kind of looks like there's a little hole here. Like these are like transport proteins in a way. Not really though, but they're just open gaps that's established by those proteins. So we say that those are the gap junctions. But of course, in plants, we call them something else. I know this seems like a minute detail, but whenever I see an essay question and it makes you have to know something, I always make sure that I teach it to you. So gap junctions in plant cells are not called gap junctions. They're called plasmodesmata. Plasmodesmata. And that's what it's kind of showing you here. The fact that we have these gaps where substances, cytoplasm, water, glucose can flow from one cell to the other. It's through those gap junctions, we call them plasmodesmata. Hey, at least it starts with PLA, kind of like what like plant does, okay? So that might help you remember plasmodesmata. That's one little small piece of information, but if students didn't know that on a test one year, like then they missed a point, okay? All right. Now, the whole rest of this worksheet is over the last part of the lecture, which is over signal transduction pathways. If you have not watched that lecture, when I give you your assignment today, you are probably going to have to go watch that lecture because you're not going to be able to do it. I kind of warned you about it. I said you're not going to be able to do much today if you haven't watched that lecture first, okay? So before we answer any questions here, I'm going to show you the basics of a signal transduction pathway. Note, what you are going to have to do is design a model of a cell, or you can use the one I designed, or you can draw this out, but ultimately you're going to record yourself explaining a model of a signal transduction pathway. Now, the one that you're going to explain is going to explain how your liver cells receive a signal, and then that causes them to release all this glucose into your body. Why? Because there's like a bear that's about ready to chase you, okay? You're scared. So you need a lot of glucose in your system so that your cells can generate ATP and you can either fight or you can flight, okay? This model that I'm showing you now is not that same model. It's a different one, but it is a great graphic of basics you need to know about any signal transduction pathway, okay? So let me go to the very beginning here. Okay. With your fingers, you're going to show me a number. How many main parts are there to a signal transduction pathway? I can tell the people that watch the lecture. Okay. There are 3 parts to a signal transduction pathway. You have to be able to list them and tell me what happens in each. First of all, reception of a signal. Cells don't do things until they receive signals to do so, okay? So what we just watched here, let's watch it again, is this cell is receiving a signal. The signal is a hormone. It's that yellow circle. By the way, you should be able to tell me, is that yellow circle a protein hormone or a lipid hormone? That's review. Lipids can cross lipids. The lipid hormones would be able to come right in and bind to an intracellular receptor. That hormone must be a protein. It's large and it's charged and it's not just coming right through that membrane, okay? So it hooks onto a receptor protein. That protein is never going to come into the cell. I have people that'll tell me that like insulin hooks on a receptor protein and the receptor protein brings it into the cell. No, the receptor protein is not a transport protein. Transport proteins bring things across the membrane. That protein-based hormone is just going to hook on and it's going to activate molecules in the cell, okay? So that's reception. Whenever a hormone binds to a receptor protein. All right, now let's watch what's going to happen. When that happens, the receptor protein changes shape. Watch. That shape change allows it to do something now different that it couldn't do a little bit ago, which is to activate another molecule that's in the cytoplasm, okay? When that molecule is activated, the only thing that it can do, the only job it has is to activate another molecule. This is a huge idea. I'm going to play that again because RAF is now activated and you should be able to tell me why it's activated or how it became activated. So watch. So everyone open chat for me again. Open chat for me. I'm going to play it again and you're going to answer, how was RAF altered so that it became activated? How was RAF altered so it's now active? You see that? Okay, what changes happened there? I'll do it one more time. Okay, so in your chat, go ahead and tell me, and you don't have to push submit yet. We'll do it on 3. How was RAF altered by RAS? Okay. All right, go ahead and push enter for me. Awesome. Okay, so RAS is an enzyme. It has an active site. And when it's active, RAF can fit in the active site of RAS, and so can ATP. That's another important thing. You all got that this phosphate was added, that P was added. You need to tell me what that is. That is a phosphate group. And you also need to tell me where it came from. ATP. How many phosphates does ATP have? 3. So RAS is able to take a phosphate off of ATP and stick it onto RAF. And that's how ATP is used for energy. I know you guys have this other idea when we say that ATP is used for energy, like you think a phosphate comes off and like this energy just blows up and out, and somehow the cell's able to use that released energy. That's not what happens at all. How ATP is used for energy is when a phosphate is removed from it and that phosphate is put onto a different molecule, it changes the shape of that molecule and it now can do something that it couldn't do, okay, before it had that shape change. That's the only way that ATP is used in the cell. It provides a phosphate, okay? Now, we're looking at right now, what this is called transduction. The signal is being spread from one molecule to another. Transduction is kind of like the spreading of a rumor. So when you tell your BFF a secret and you say, don't tell anybody, you know that they're going to go tell like 12 people like in the next 2 minutes. And then those people are going to go tell other people in the next 2 minutes. And in like an hour, the entire school knows your secret. Okay. That's the transduction of the rumor or the transduction of the signal. That's a second part to a signal transduction pathway. Okay. So let's keep watching. Now that RAF is activated, it can do its job. Okay, you see what it did? And guess what? It had to use an ATP molecule, and it took a phosphate off of the ATP and put it on MEC, whatever that is, just an abbreviation for protein. By the way, these are all proteins. They were made by what type of ribosome? Go ahead and open your chat for me. Don't hit enter until I say so. These are all proteins sitting in the cytoplasm off until they get turned on. What type of ribosome made them? We have 2 types of ribosomes. Okay, go ahead, hit your enter. Yay, you guys actually listened to me. Have I mentioned that this is the strongest group of AP students I've ever had? In my history of teaching AP, I'm not blowing smoke at you. You guys have been thoroughly impressing me. So keep up the good work, all right? Okay, let's keep watching. So this is still transduction. Okay, now everyone find MEC, M-E-K. It's the light blue one, okay? If MEC is left on, then it's just gonna keep turning on ERK, turning them on, turning them on, turning them on. Now, it's just like with other biochemical pathways. We have to be able to shut them down somehow when we don't need them to keep activating what they're supposed to activate or doing what they're supposed to be doing, okay? Watch MEC. Watch how we inactivate it. PTase, you know it's an enzyme, right? Because it ends in ase. Watch what it does. How did it inactivate it? Took off that phosphate. Watch again. Its phosphate's gone, okay? So now it's inactive. MEC is turned off, all right? But these are turned on and the signal is continuing to be spread. We're gonna learn more about this in like February. You're gonna learn about the promoter and what these things are. These are transcription factors. They were off, but now they're on and now transcription can take place, okay? Once all those things are in place. And then we have RNA polymerase make an mRNA. This should make sense to you. Out comes the mRNA. The mRNA is going to go to a ribosome. And now this cell is going to make a protein that it wasn't making 5 minutes ago. All because it received a signal to do so. That signal needed this cell to produce a certain protein so it could do a certain job for the organism. Okay? All right. Couple things. Do you see how efficient it is to have proteins made and ready to work, but they're just off? Okay, does that make sense? That's very efficient. If these are all proteins and the cell had to first make them all after the receptor protein hooked on, that would take way too long for your cells to respond. Okay, so that's something else that we see in cells. We have all these proteins that are there, but they can be turned on and they can be turned off whenever they're quickly needed. Okay, all right. Couple other things I want to point out on this worksheet, right? And then we're going to get into what the models that you guys are going to do today, okay? Let's flip over to the back here, and I'm going to jump on down to this very last question, okay? Now, by the way, this one is showing you that's the hormone and it is hooked on to a receptor protein. It says signal, it says receptor. So that must be the receptor protein, okay? Now, the first molecule that usually gets activated in the transduction part to spread the signal is CAMP, cyclic AMP. But one thing I want to show you here, the model of mine that you're about ready to watch, I just show one CAMP getting activated by the receptor protein. That's not true. This receptor protein can activate a ton of CAMPs, okay? This is like your best friend going and telling your secret to 10 people, okay? And then if each of them can go and activate 10 people or tell 10 people, do you see how your rumor grows or spreads very, very quickly, okay? And that's what happens in the transduction part of signal transduction pathway, okay? This is called a cascade. You guys know what a champagne cascade is? I don't know if you've ever seen one like at a wedding or something fancy that you maybe been to here. See if I can open this up real quick. Okay. So this is a champagne cascade where they stack the glasses and you pour the champagne here in the top and it activates 4 glasses. Then those 4 glasses activate like 12. And then those 12 activate like 24. So this is called a champagne cascade. In regards to signal transduction pathway, we can call that a signaling cascade. If a cascade is happening, the signaling cascade, if a cascade is happening, then the signal is growing, or we say that amplification of the signal is taking place. That's a word that you need to know. The signal becomes amplified when one molecule activates many in the signal transduction process, okay? So that's like an amplifier. It takes a guitar and it makes the sound big. This one little step right here can lead to the growing of the signal or the amplification of the signal. So then it spreads very, very quickly if one can activate several, okay, or amplify the signal. All right. Since I have you guys on here now, let's talk about your homework assignment, okay? So I have to do what is good for you guys and not necessarily what is easy for you. And so I don't have time to give you the cell unit quiz 1 in class because I have to get through photosynthesis and respiration before Christmas or it's not going to happen. And like I said, you guys are a strong class. And I firmly believe you guys, regardless of what's been happening with the pandemic, that you're going to knock that test out of the water. Okay. So I'm going to issue a at-home test to be taken over the weekend. It is in Canvas. If you click on assignments where you guys normally go, then you're going to see you guys have been assigned Form B. And it really is the only one that you should be able to even see, okay? Because I didn't assign you guys Form A. So you guys are going to get Form B, and it's timed. It's 50 minutes. If you do not study at all and you think, oh, I'm just going to look up all the answers as I go, you are probably going to get halfway done because it will shut down in 50 minutes. So this is forcing you guys to go and take a second to study lectures 1 through 4. If I just give you a Unit 3 test at the very end, it's not going to go well. You need a pause. You need to study all the stuff that I've thrown at you in the first 4 lectures. And then we'll add a couple more lectures for a unit test, okay? So that is due by Monday. I think it says that it's due by like midnight on Sunday. I will send you guys out a reminder. Now, to help you prepare for that, remember there's learning targets. Also, if you click on quizzes, scroll down to the bottom. I do have a practice quiz ready to go that has multiple choice grid-ins. Grid-ins are usually the number questions. Think lecture 2. Surface area, volume, surface to volume ratio, diffusion rate. I made you guys calculate all that stuff. Those are what those questions are going to be like. At the very, very bottom is where you guys can find practice quizzes. So click on quizzes, go all the way down to the bottom. I have one right there for you. Unit 3, cells practice. And I have 5 essay questions it's going to pull. And again, you can see all your answers and scoring guides and stuff like that. Okay. All right. So again, I can't say no notes. You can have your notes out, but if you go to look up more than a few answers, you are never going to finish. And that's what happened at the AP test in May. You guys could use your notes, but ain't nobody getting finished if you use your notes. My kids said we didn't have time at all to even look at any note packets that we had out. They just said it would take too long to find the information. So you either know it or you don't. Okay, so make sure you guys study before. All right. I also have a lecture that is due by Monday. It is the 5th lecture. I have it right here is the link for it. I'll tell you this, it's going to be a review. So you have my permission to watch it on time and a half because it's going to be over diffusion, osmosis, facilitated diffusion, active transport, endocytosis, exocytosis, and tonicity, hypertonic, hypotonic, isotonic solutions. Taught all that stuff in honors biology. The only thing that's going to be additional is maybe talking about like contractile vacuoles and water potential. But lecture 6 is all about water potential, to make sure you guys know that. So that's the addition here at the AP level when it comes to transports. You're going to do a virtual lab on Monday. I'll tell you this, if you don't have the lecture watch, you still will probably be fine doing your virtual lab, unlike today. Because if you haven't watched that lecture, you're not going to be able to do what I'm going to ask you to do, and you're probably going to have to go watch it, okay? All right, so speaking of, everyone make your way to Google Classroom because you guys are going to open a video and watch it on your own, whereas I am describing the model and you are going to do something like I do, okay? So go ahead and go to Google Classroom, click on Friday, and there's a video here that says STP modeled, okay? Before you guys hit start, note, you can use this model and explain it and record it, okay? So you can use my model. I made one for you to use. I thought that'd be easier than you making your own, okay? So you can use this or you can draw it out or whatever, but keep this in mind. This is the signal transduction pathway I'm going to have you explain. And if you can explain one, then you can usually figure out all of the other 500,000 ones that they could possibly show you on the AP exam, okay? So click on that and watch that video. When you're done, put your hand icon up. Also, if you think you want to use my model, I have it here, but there's a learning curve. You guys have to learn how to click on molecules and like move them and drag them around and group them and ungroup them. So play around with this to see if this is something you want to use, or you're like, forget that, I'm just going to draw it, Miss Spencer, and explain it, okay? All right, so I'll be quiet. You click on that video right there and you watch it and really dial in.